Caroline Gluck. I'm a pediatric nephrologist at Nemours AI DuPont Hospital for Children. This is my first presentation at the Junior Investigators Network, and my talk is entitled Nephrotic Syndrome, the Right Medicine the First Time. The nephrotic syndrome is characterized by massive proteinuria, hypoalbuminemia, and as a result, children become very edematous. They have swelling often in their bellies, in their legs, around their eyes. They're just very puffy, and they also have hyperlipidemia. It's a rare disease, and despite that, it is a leading cause of acquired end-stage renal disease in children requiring dialysis and kidney transplantation. It does have many complications. Top among them are complications related to fluid overload, pleural effusions, ascites, infections of that ascites because these children are actually immunocompromised. Between the hyperlipidemia and the loss of coagulation factors, these children are very hypercoagulable, and they have thrombosis. They can even have strokes. As we already mentioned, it is a leading cause of renal insufficiency. And even after a kidney transplant, this disease can recur, which can be really devastating. It can occur within hours and days of the kidney transplant. It is a disease of the glomerulus. So your kidney is made up of approximately 1 million glomeruli each. And that's the filtering unit of the kidney. The blood goes into the glomerulus, gets filtered into Bowman's capsule, and becomes the ultrafiltrate. And it gets filtered through the slit diaphragm in between podocyte foot processes. So you can see these interdigitated foot processes of the podocyte cell, and that's actual filter of the kidney, and that is what actually becomes defective in children with nephrotic syndrome. We classify nephrotic syndrome mainly because we don't fully understand the pathophysiology of nephrotic syndrome, and so we classify it by various classifiers: age. So some children present congenitally, so before 3 months of age. In infancy and childhood, there is also adult onset nephrotic syndrome. We classify it by whether or not they respond to steroids, so if they're steroid sensitive or steroid resistant. We classify it by a description of what their renal biopsy looks like. So things like minimal change disease, where on light microscopy, the kidney looks unchanged, to focal segmental glomerulosclerosis. These are descriptions. And then finally, genetic mutations have been noted in approximately 30% of children with steroid-resistant nephrotic syndrome. And so, if they have a genetic mutation, they'll be characterized by that. But the correlation among these categories is really poor and not well described. And despite all these things, the treatment for nephrotic syndrome is really empiric. So as long as it's not congenital nephrotic syndrome, children are typically treated with high-dose steroids for somewhere between 4 to 6 weeks. And if they respond, fantastic, they're steroid sensitive. But even despite that, they might relapse. And the majority of children will relapse when they have colds or things of that nature. And then about 30% will be steroid resistant. And for those children, they're started on ACE inhibitors and ARBs. And they're, again, empirically started typically on calcineurin inhibitors for approximately 6 months. And this is despite what their biopsy shows. And if they achieve partial or complete remission, then great. If they don't, they're trialed on mycophenolate. All of this is really empiric, and for the children who are not responding, they're still very swollen. They're still experiencing all of the side effects of the nephrotic syndrome itself, as well as side effects related to the treatment, which are, in general, broad spectrum immunosuppressive medications. What we do know is that if they respond, if they're able to enter full remission, and this data is taken from the PODONET registry in Europe, they do better. So the renal survival for the children in green who are in full remission is better than those who have partial remission in yellow. And both are better than children who don't respond at all. And we also know that children with genetic forms of nephrotic syndrome do worse. And so on this graph, the genetic syndrome, nephrotic syndromes are in blue, and you can see they have really poor renal survival. The majority of genetic causes of nephrotic syndrome affect that podocyte cell that's creating the filter for the kidney. And this is a cartoon of two of those podocyte cells next to each other. And in between them, they're forming the split diaphragm, which is really created by nephrin and podocin. And you can see that there are a lot of other structural proteins, mainly that contribute to the formation of that filter. And studies have shown that mutations in these different proteins can lead to nephrotic syndrome. But aside from nephrin, so the one that's really in the middle, creating that bridge between the cells, other mutations really present at variable ages. And so this was one of the largest studies published in 2018, showing that really, aside from nephrin, which you can see is the red genetic mutation in that graph that presents in most part as a congenital nephrotic syndrome, so before 3 months of age. A lot of these mutations are showing up at different age groups. And so children might not present until they're 18, might not even present until they're into adulthood. And other studies have shown that while patients with a nephrin mutation are really unlikely to respond to any immunosuppression, these other mutations may or may not respond to therapy. And so most likely there is some kind of complex genetics that is happening here that is not being fully explained by the monogenic mutation. And so that's really part of what we're interested in looking into. Because this is a rare disease, we really need large patient populations in order to study it. And so we're working on two complementary projects. The first project is a collaboration with Dr. Anya Kozel at King's College in London. And through this collaboration, we have access to more than 600 children with steroid-resistant nephrotic syndrome who have a whole exome sequencing and deep clinical phenotyping in terms of their response to medications. We're additionally going to be enrolling approximately 160 children at Nemours with nephrotic syndrome, utilizing the Nemours Biobank in order to complete this project. The second project is using electronic health records data through something called PedsNet, which currently holds data for more than 6.5 million children across 8 children's hospitals. And there's already been a computable phenotype algorithm that's been created to identify cohorts of children within PedsNet who have nephrotic syndrome. So I'll talk about the whole exome sequencing project first. Again, in London, we have about 600 children with steroid-resistant nephrotic syndrome, and at Nemours, we're going to enroll about 160 children with steroid-resistant nephrotic syndrome. The idea is to use whole exome analysis to not only identify mutations in the more than 53 genes that have already been associated with steroid-resistant nephrotic syndrome, but also to look across the entire genome for other signals which may either be associated with steroid resistance or associated with response to other immunosuppressant medications. We're going to look at complex genomic lesions such as copy number variation, which has really not been well studied in this disease. We'll look across genes that are known to be influenced by steroids and other diseases, SNPs in these genes, both in the coding region and in non-coding regions that might affect things like gene expression, have been shown to affect therapeutic responsiveness in diseases like rheumatoid arthritis and asthma. There's also some signals that have shown up in HLA proteins, which is interesting because, as I kind of mentioned earlier, infection tends to cause these children to relapse. And so there is some immune component to it. And so the goal is to take all of these biologically significant signals and feed those into basically a classifier system. And so this is just a cartoon to give you an idea that you may have a patient who exhibits different symptoms, might have different signals across their genome. They might have a certain genetic mutation in that podocyte cell. They might have a copy number variation, perhaps affecting one of those genes in the podocyte cell, or potentially a gene that affects responsiveness to steroids. They might have a SNP in a location of that HLA, and this is some preliminary data from the London cohort that you're seeing here. And we'll take all of that information, those different features, and use mainly random forest, but we're going to try other classifiers using machine learning to really identify, which patient's genomic makeup can predict response to certain medications. And to really create a prediction classifier, we'll use a portion of the London cohort, so a portion of those 600 samples to create this. We'll test it on approximately 20% of that cohort. And then we're actually going to test the classifier in the Nemours cohort. So we'll have a development, a test, and a validation phase to this study. The second study, as I kind of alluded to, is leveraging electronic health records data. There is already a validated computable phenotype algorithm to identify patients with nephrotic syndrome across the PedsNet system. And we've identified approximately 3,000 patients across the system, which is pretty amazing considering how rare of a disease this is. And our plan is to narrow it to the steroid-resistant population and identify those who've potentially had genetic testing done for clinical reasons. And so many nephrologists order genetic testing. These panels are available. And nobody's ever really collated all of those results. And with the genetic testing results and the EHR clinical phenotype, we're hoping to have another means to better classify these children and identify correlations between genetic testing results and therapeutic response and outcome. So that study's just a little bit further along potentially, and we've identified a cohort at Nemours with steroid-resistant nephrotic syndrome, about 217 children, and about 85 of those children have proteins that are likely indicative of genetic testing. Those children had a higher incidence of requiring dialysis and kidney transplantation as opposed to the general nephrotic population. And our next step is really to start chart reviewing these children and to identify the genetic testing results. Once we've kind of validated this process at Nemours, our plan will be to expand across all 8 of the PedsNet centers and gather all of that data. So in conclusion, we need large cohorts of children and complementary data sources to better classify children with nephrotic syndrome, with the idea of really using big data, machine learning techniques to create a novel classifier and really better treat these children the first time rather than having this trial and error methodology where the children are kind of suffering along the way. Obviously, this is a very large project and it requires many different people. I have a wonderful mentoring team at Nemours and across different networks. So Erin Krauge is my mentor at Nemours, and Claudine Yerkovitz is my mentor at Christiana Care. And they're helping with the genomics and electronic health records projects respectively. I mentioned Dr. Anya Keisel at King's College in London. Michelle Denberg is up at CHOP and has been very instrumental with the PedsNet project. Anne-Marie Brescia at Nemours is my faculty mentor. Carl Franke at Nemours is helping with the genetic analysis, and just freshly recruited Mauricio Ferrato and Dr. Sunita Chandrasekaran at the University of Delaware will be helping with some of the machine learning aspects of this project. Dan Eckridge and Suzanne McCann at Nemours have been helping with the PedsNet project, and finally Marley will be helping with the biobank recruitment of subjects. This is my newest colleague, Rupert, who has been diligently sleeping while I work from home. This work has been supported by the Mentored Research Development Award through ASL, and I recently submitted to the Delaware CTRSL Pilot Grant, as well as the Neptune Career Development Fellowship, with upcoming submissions planned for the CHOP Pediatric Center for Excellence Pilot and Feasibility Grant. And that's all I've got, so I'm happy to take questions.